Bismillahir Rahmanir Rahim. Good morning, audience everywhere. Today we are going to start chapter 2, which is about the cytology or the science of the cell. Cytology is a terminology that is formed of two words, cyto and ology. Cyto is the Greek Latin words, means cell, while ology means science. So cytology is the science of the cell. Cytologist is the scientist or the biologist who deals with cells in his lab or his researches. By the word cell, we do not mean the cell phone nor the detention cell in jail. What we mean is that the cell is the smallest structural and functional unit of the organism. That's to say, it is of life. It is the smallest structural and functional unit of living cells and of life too. Dealing with cells and dealing with biology, we are going to use our textbook here, textbook of biology. This is the textbook that, or the course notes or course book that have been recommended by the Board of Education in Cambridge University, that's responsible for the ICGSE in its post version 0610 or 0970, and also of the O-level, Cambridge O-level biology. So we will deal with this book, and it is here. It is the fourth edition, the same authors of the third books, but here there is a lot of there are a lot of topics that have been removed from this one. Let us go further and see what we are going to do with this cell. In our study of the cytology, we have to deal with what we have been known the cell theory. The cell theory is the theory that telling us something about the cell. The terminology itself is not found in your book, but the pillars of the four pillars of this theory are there. So I just package them together and name them the cell theory as old textbook known it like that. So we know that the cell is the smallest structural and functional unit of the organism or of the living organism or of the life. So this based on four pillars. Number one, that all organisms are made up of one, that means one cell, and in this case we name it unicellular, like the bacteria and yeast. So this is a unicellular organism, or more than one cell. That means it has many cells or multiple cells. So it's called multicellular. An example of this is mammals and birds. Even the plants are the same of that. The cell is the basic. The second pillar is that the cell is the basic unit of structure and function of organism, and this is what we already said before. New cells arise only from existing cells by cell reproduction or cell division. So there is no cells come from nothing. All cells are built up or exist as the daughter cells of pre-existing one, and this is a most important criterion of the cell theory. Cells exchange substances with their environment by transporting these substances in and out of the cell across the plasma membrane, and we are going to deal with this part a lot because it is one of the most important characteristics present in living cells. Let us go forward. Before going further in cytology, we would like now to classify cells based on a rule, a concrete thing that makes real differences or make some sort of perfect classification. The first type of classifications we are going to take is the classification based on the development of the nucleus of each cell. This type of classification that based on the nucleus divide or classify cells into two categories. The first category is called prokaryotic. The second one is called eukaryotic. So pro means at the beginning or primitive or before, and karyot means nucleus. Eukaryotic, eu means true. Karyot means nucleus. That means those are the cells with or contain a real and true nucleus. Based on this and other criteria, we divide cells into prokaryotic cells and eukaryotic cells. So this classification mainly based on the development of their nucleus. So based on the nucleus, we say that in the prokaryotes, it is a primitive or none. That means it may be not present either, and the nucleus is just a nucleoid region, just the region that contain circular DNA. While this is something different in the eukaryotic, the more developed nucleus, where the nucleus contain well-developed nucleus, and also the DNA is protected by a nuclear envelope. That means it protected by a sheath that harbor or limit the shape of the nucleus, and also it had a nuclear pores, that this nuclear envelope, and by pores means holes that are provided to permit into and out a movement of substances into and out the nucleus. And also the DNA here is different from the DNA of the prokaryotes also, or in primitive cells, that it's not a circular, it is a linear one. There are other criteria like presence of cell wall, and this is present, and eukaryotes present in prokaryotes. It's not present in animal eukaryote, but present in plant eukaryotes in eukaryotic cells. Membrane-bound organelles, and we're going to talk about this when we come to the organelles, is absent, and it is present in the eukaryotics. Histones and chromosomes are absent and present in eukaryotic. I just want to define organelles. Organelles means small organ or tiny organ. And whenever you find absent, you are going to find it in red color just to make you easily memorize what I'm telling you about. The example of prokaryotic cells like the bacteria, very good example, and the plant and animal cells in general are examples of the eukaryotic cells. Another classification is very important because it is the main classification of your textbook or your course book, is the classification based on the origin. From where did this cell come from? And we have two main categories based on the origin, either an animal cell or a plant cell. And here we are going to see them in these two sketches based on the visualization of the animal and plant cells under light microscope. And I want you to put 10 red lines under the light microscope, because that means you cannot see them by the naked eye, but also you cannot see more than them using the light microscope. If we want to go for further details, and we are going to talk about this when we talk about the microscope in the last lecture. But here, the light microscope is very important here, and those things are important to use them under light microscope. What we are going to see here, it is just under light microscope. And why I'm saying 10 lines, because always it comes in the test, on the exam. You are going to see that. Now, I also want you to recognize this rule of 3 by 3. Why I'm saying this? Because we have 3 similarities and 3 differences. And based on this, it is 3 by 3. So there is a cell membrane in both of them here, the inside one. And the cell membrane is also called the plasma or the surface membrane. Also, both of them has a nucleus. Each of them has a nucleus. And each of them has a cytoplasm. So those 3 are similarities. Those 3 are the similarities between them. They are found in both of them. I have also 3 differences between that are present only on the plant cell, but not in the animal cell. What are they? Those are the cell wall, the large vacuole, and the chloroplast. Those 3 are not like those 3. And this is why I am putting them here in green color. That 3 by 3, one of them is for both of them, but the other one is only for the plant, and this is why I'm doing here in green, in order you to be sure that you have to know that. So those are differences. Those are only present here. Now, on the moral of the story, you now have 2 types of classification for the cells. One based on the development, how the nucleus of the cell is developed, and the other one based on the origin. From where did this cell come from? Either from an animal cell or a plant cell. We are going further to discuss more details about these things. Moving forward, and we still visualizing those cells under light microscope, and I warned you about the light microscope. We are going to see this picture. Now, those are the similarities. The 3 violet ones, they are present in both animal and plant cell. The cell membrane, the cytoplasm, the nucleus. The cell, as I told you, is also named the cell wall or the cell membrane is also called the plasma membrane or the surface membrane, and it is present in both of them. It is just a thin layer of protein sandwiched by 2 layers of fat. And also it is porous, that means has many pores membrane, and also it pressed tightly in plant cell to the cell wall, because this is characteristic for the plant cell. The cytoplasm is present in both cells, and it is just a clear jelly or colloid substance. 70% of it is water and 30% of it is salts and other dissolved material, especially proteins, and this is why it's colloidal or jelly-like a structure. The nucleus is large and present in pores first, present in pores and surrounded by a double membrane studded by pores. It harbors the nucleolus. The nucleolus is not found in your book, but it is a little structure present inside the nucleus and contain also the chromosomes, and the chromosomes are made of DNA. This is all about them. Do not forget here the rule of 3 by 3, and do not forget again that this is structure based on visualizing those under light microscope, not electron microscope. In this part, we are gonna continue comparing the three similarities: the cell membrane, the cytoplasms, the nucleus, from the function point of view. And since they are the same, they have the same function in each cell, whether it is an animal or plant cell, because it's present in both of them. So the most important two functions for the cell plasma or surface membrane is the protection, protect the cell and separate the internal and external environments. And also it has selective permeability. It's a very important characteristic. That means it is partially permeable. That means it allows the passage of some substances and molecule to pass through it in and out the cell. Again, it means transportation of materials entering and exiting the cell. So it has those two things, and it is very important to know them. And I also want you to remember that the structure matches and function. This is our second part, which is very important. The structure matches the function. The second rule that we have it, and already we have taken it last time. Cytoplasm is present in both cells. It is the site of the cell metabolic reaction. That means the sites of chemical reactions of life, the chemical reaction that present in living organism. And also it harbors or contain the organelles of the cell. Nucleus, it is present in both of them, and it is developed nucleus. That means both of them are eukaryotes, and it is the sites of genetic information, DNA plus chromosomes. Also, it is the sites of RNA synthesis, transcriptions, and we're going to talk about this when we come to transcription. And also it is important for synthesis of right source of proteins, because each organism has a specific proteins to deal with. The last but not least, it is responsible for cell division. Stepping forward, still we are under microscope. We in the last scene or in the last slide, we have studied the or explore the structure of the and function of the similarities. Now we are going for the differences. The differences are restricted for the plant cell, and this is why we have 3 by 3 rule, where this 3 is in green, and here every present is in green. So all these structures and features are present in the plant cell, not in the animal cell. The cell wall, the chloroplast, the vacuoles are specific for the plant cell. They are differences that present there, and we have to know that we can see them under microscope, under light microscope, and this is very important because in the question you may see light microscope. You are not, it is wrong if you write something, if the question is about light microscope, it is wrong if you answer something that is related to electron microscope. Now, for the cell wall, it is present in the plant cell nor the animal cell. It is rigid and made of cellulose, which is a complex polysaccharide compound present in the cell. It is outer cell frame. It is the outer cell frame and resemble the outer skeleton of the cell. It makes the cell with a specific shape, and this is one of the differences between the plant cell and the animal cell, that the plant cell has a specific shape, regular shape, while the animal cell hasn't. It has no regular shape. Cytoplasm, chloroplast is present in the plant cell, but not the animal cell. It contains green pigment called the chlorophyll, with starch and grains. Chlorophyll is a terminology that made of two words, chloro and phyll. Chloro means color, phyll means love, so this is the organelle that is loving color, and it is a green color here, so this is why it is chlorophyll. And it has a double membrane bound organelle and contains thylakoid. Thylakoids are like the coins. You see, when you put the coins together, it makes a stalk, and this stalk is called, or stacked coins, to increase the surface, because any additional something that structure that present in any surface, it increases the surface area, and this is for a reason. Some animals may have contain tiny granules which are not chloroplast. They called glycogen granules, and this glycogen is a complex carbohydrate. Especially carbohydrate, specifically present in the animal cell, not in the plant cell. The last one is the large vacuoles. It present in the plant cell, not the animal cell. It is a fluid-filled permanent space surrounded by a single membrane. It has no dipole membrane. It contains a blend. Blend means a mixture of sugars and other substances called sap. And animal cells may contain much smaller membrane bound containing a space, mostly contains solution, food solutions. We are still working with the similarities and dissimilarities, or unsimilar differences between the animal cell and plant cell, and now we are going to see the function of the differences. We have three features that are differ between the two cells, and those are the cell wall, the chloroplast, as well as the large vacuole. The cell wall is absent in the animal cell, present in the plant cell. It provides tensile strength support and protection against mechanical and osmotic stress, and this is very important. We are going to deal with this a lot of time because there are very nice questions in this. This also allows cells to develop turgor pressure. That means the pressure of the cell contents against the wall, and also provides regular shape to the plant cell compared to the irregular shape of the animal cell because the animal cell is deprived of the cell wall. Chloroplasts are also absent in animal cell and present in the plant cell. It is the site of photosynthesis, which absorbs the light energy from the sun and convert it into chemical energy. That means it is converted into food. Also, it is the site where the storage of starch, grains, and fats takes place. The last but not least is the large vacuole, which is again absent in the animal cell and present in the plant cell. The large vacuole provide turgor pressure for gross plant support and also storage of substances. Example is the sap. You know that the sap is a blend of sugars and salts and other materials. I insist to you to remember the two rules that we have already studied in this lecture. The first one is you have to remember all the time the structure matches the function. It is a very interesting and very important rule in biology. The second rule for comparing the animal to plant cell is the 3 by 3 rule, that they have three similarities, three features that are similar in both, and three other features that are different. They are only for the plant cell. Let us go forward. This slide is only a demonstration. It is a demonstration for the future work. What I am going to do is to reorganize all the past papers, and I'm going to divide them into three main folders. The MCQ folders, that means the paper 1 for the core and paper 2 for the extended together, and then paper 3 and paper 4 together, and paper 5 and paper 6 together. And what I'm planning to do is to make a folder that answer all these questions and also answers with explanation why this is the correct answer, why it is the right answer. Also, I'm going to have a specific lecture for those questions and those files. So do not panic. I'm going to do the homework that I have to do, and you are going to find out a good solution for your examination. Thank you for having me all this lecture. Thank you also for tolerating me. I hope that it is an enjoyable class. It was my pleasure to have you all. And what I would like to say, I'm waiting to hear from you again and see you again. Thank you. Have a good day and bye-bye.